Hello, my name is Kathy Singh, and right now I'm pursuing my Master's of Zoology from Maharishi Dayanand Saraswati University of Ajmer. I want to become a molecular biologist. For that, I'm going to start my PhD in the upcoming year. I'm teaching from past 5 years to the AP Biology students as well as NEET aspirants who are going to write their pre-medical exam in India. I teach biology to them, and in biology, the most important topics are cell and cell cycle, which we are going to talk in detail in this class. We are going to study about cell cycle, that how the cells are going to divide. What is the process of this division? Okay, the cell cycle is a complex process in which different steps are involved. So let's start with the basic information or introduction that what is cell cycle. So what is the introduction? In a human body, there are two types of cells. First, the somatic cells, that is body cells, with the cells which are present in our entire body, in my cheek, hands, legs, stomach, intestine, every other part. And the other form is the gametes, which is going to participate during the sexual reproduction. Getting me? Male sperm and female ova is going to fuse together to form a zygote. So who is participating? Sperm and ova. And where they are formed? They are formed in our sexual organs in the testis as well as ovary. Getting me? So what is a gamete? Gamete is sperm in case of male, and in the case of female, it's egg or ova. Okay. Next is genome. Genome is a very small term for genetic makeup. Okay. So our entire body's genetic makeup is known as genome. It is a total DNA allotment, total genetic makeup. Then what is chromosome? Chromosome looks something like this. Okay, can you see? So it's a long thread. Can you see? It is a thread. Can you get me? It is a thread. It is made up of thread, but this thread is arranged in a particular pattern. Why? Because it is arranged, it's wrapped on some proteins, and those proteins are known as histones. Getting me? So this particular thread is what? It's a DNA thread arranged on a protein. Why? See, the DNA thread, if I'm going to make a DNA, my DNA thread is 2 meter long. Are you getting me? And my height is 1.65 meters. It means the DNA thread is longer than me. How we are going to arrange such a long thing in such a small thing? Cells are like so small that we cannot even see them with our naked eyes. We need a microscope to look at them. And in that cell, the chromosome is present inside a nucleus, even smaller. Getting me? This 2 meter long thread, how this 2 meter long thread is arranged in such a small cell? It is arranged in a small cell because of protein that is known as histone. Getting me? This long thread of DNA is going to wrap around these histone proteins. Then it's going to get arranged inside the nucleus. And when they're going to wrap themselves, when this long thread of DNA is going to wrap themselves on the histone, it is going to take a shape something like this, which we call chromosome. Okay. Okay. So chromosome, now in the chromosome, if my chromosome is going to divide, for example, this is made up of DNA thread, right? If my DNA thread is going to divide on the in the chromosome, what will happen? The chromosome will contain two more legs. Getting me? Two more legs and threads. Okay, this part is known as chromatid. Getting me? So it means when my DNA content is double inside a chromosome, there are two sister chromatids. Getting me? Two sister chromatids are present in a DNA. So now let's study in my mother. For example, I contain 46 chromosomes. All the body cells of my body contains 46 chromosomes. How this 46 number is maintained? How this 46 chromosome number is maintained? Let's see. In my mother, there are 46 chromosomes. In my mother also 46 chromosomes are there. And same goes for my father. My father also contains 46 chromosomes. They are going to reduce their number of chromosomes to make ova. My mother is going to reduce her 46 number of chromosomes to 23 only. And the same goes for my father. To make a sperm, my father is going to reduce his 46 chromosomes to 23. Now, the egg contains 23 and the sperm also contains 23 chromosomes. Getting me? They are going to fuse with each other to make a zygote. And now my zygote, from which I'm I'm developed, my zygote contains 46 chromosomes. Getting me? So the chromosomes which are coming from mother side, they are known as maternal chromosome. And from a father side, the chromosomes which are coming, they are known as paternal chromosome. Okay? So I contain both the parts. I contain some characteristics from my mother as well as from my father. Now we are going to study what is DNA, chromatid, centromere and kinetochore. Okay. So for example, this is my cell. Inside my cell, there is a nucleus. Nucleus is a double membrane bound structure. So I'm making a double membrane. Okay, this is what? This is the nucleus present inside my cell. Inside this nucleus, there is a thread-like structure. Can you see? This thread-like structure is known as chromatin. This chromatin material is going to condense. It's going to go through a process known as condensation. What is condensation? Condensation is like, thickening of this wool-like structure. Can you see? Right now, particular structures, we cannot see any particular structure in this chromatid. It's just like a wool. Getting me? So these threads are going to become thick. So we can see them under the microscope. We can visualize them under the microscope. So this condensation is what? Condensation is the thickening of, it's a process in which the chromatin is going to become thick. Okay. It's a thickening of chromatin. Once the chromatin is going to get thickened, it will form a structure something like this. And this is what? This is a chromosome. Okay. And we all know that this particular thread of chromosome, it is made up of DNA. Right? DNA is going to make the chromosome. Right? So this whole DNA is not functional, okay? Most of the part of the DNA is not functional. Only a particular part of a DNA is functional, which we called gene. Gene is actually the part of DNA which is going to go. It's the smallest structural and functional unit of inheritance. What is gene? Gene is present on DNA and it is the smallest functional unit of inheritance. Okay. Now, if you're going to look, now let's come again to the chromosome. Chromosome contains a disc-like structure in the center. Okay, so this disc-like structure is known as centromere. Okay, centromere contains kinetochore on the lateral side. So what is my lateral side? The sides of my body. Getting me? It contains kinetochore. Can you see this part? This part of centromere is known as kinetochore. Now, what is kinetochore? Kinetochore is important. Kinetochore is a protein on which spindle fibers are going to get attached. Okay, I can show you the structure. For example, yes, so look at this chromosome. This is the chromosome, and inside this there's a disc-like structure, as you can see. And in this disc, can you see the structure? This is kinetochore, on which the spindle fibers are getting attached. During cell division, the spindle fibers are going to form, which is going to divide the chromosome, and the spindle fibers are going to get attached on the kinetochores of the chromosome. So let's move forward. In cell cycle, two processes are important. First process is karyokinesis. Karyokinesis. In this, karyon means nucleus. And kinesis means divide. Okay. The next step is cytokinesis. These two processes are important in the case of cell cycles. Cytokinesis. Cyto means cytoplasm. And kinesis means divide. So in cell cycle, what is important? First, the nucleus is going to divide, and then finally the cytoplasm will divide. Let's see. For example, this is my cell which contains a nucleus. First, what will happen? The karyokinesis will take place. It means this cell nucleus is going to get divided. Can you see? And finally, my cell contains two nucleuses. And then the cytoplasm will divide. And finally, two daughter cells are formed, each containing their own nucleus. Getting me? So first what will happen? This nucleus is going to divide inside a cell, and then finally the cytoplasm will also divide. During cell cycle, see, if you're going to do something, you need to prepare yourself, right? For example, if you're going to do exercise and stuff, you need to eat food. You need to grow in size. You need to make certain proteins for the process which we need. Getting me? So during the cell cycle, two phases are important. First is the interphase, and the second one is M phase. During interphase, the cell is going to prepare itself. Okay, what is interphase? It's a preparing time. And this cell is preparing itself to divide. So what is going to happen in the G phase, in interphase, three phases are there. First is G1, second is S phase, and the last one is G2 phase. What happens in interphase? That the cell is preparing. So 90% of the cell's life goes in the preparation of division. Getting me? And the M phase is just 10%. It's very it's like it's the smallest part of the cell division. The actual division happens in M phase. The actual karyokinesis and cytokinesis occurs in the M phase. But most of the cell is going to give its time to interphase, where it's not dividing, it's just preparing. It's making protein, growing in size. So what happens in G1? G1 is like growth phase, in which cell is going to grow. The cytoplasm is going to become thick. The cell membrane is going to become thin, so they can divide. Okay. And in S phase, the DNA amount is going to become double. Okay, the DNA's amount is going to become double. So how it's going to become double? For example, this is my chromosome. The chromosome is going to divide. See, I am doubling the amount of DNA, not the chromosome. This is a one chromosome. When my DNA is going to replicate, or when my DNA is going to synthesize, two chromatids are going to form. Can you see? It is again only one chromosome with double the amount of DNA. The amount of DNA is doubled, but the chromosome number is still one. Okay, how many chromosome is this? This is only one with the DNA which is doubled. Okay, the amount of DNA is doubled. It contains two DNA, double DNA, but chromosome is still one. Getting me? In the G2 phase, again some proteins are going to form, which is like very important during the cell division, and spindle fibers are formed. Spindle fibers are formed from the centriole. You must know a structure like this. Centrosome, centriole. Centriole is going to make spindle fibers, which is going to attach to the kinetochore. Remember we studied about the kinetochore? So see, cell cycles, interphase. Interphase is G1, S and G2. What happens in gap1 phase? Growth. Cell is going to grow through protein synthesis, and the cytoplasm is going to become thick. Okay, then comes S phase, that is synthesis phase, in which DNA's amount is going to get double. Getting me? But the chromosome number remains same. You can write chromosome number remains same. Okay. Next comes G2 phase, that is gap2 phase, in which again the growth is happening. Proteins are going to synthesize, which we need for the division, and the spindles is produced. What is the most important part of gap2 phase? That spindles are produced. Next is M phase. In this, nucleus is going to divide. That is known as karyokinesis. Okay, and cytokinesis will also take place. That is the cell division. Okay. Now, in the cell cycle, when we are going to dwell in the cell cycle G1, S, and G2, it's a long process. So, we will study these topics in the upcoming class. I hope you like this demo and my teaching methods. I'm looking forward to work in your company. Thank you and have a nice day.